started again. I'm just going to briefly introduce the next introduction for our speaker. And this is Dr. Andrew Heyman, who is the director of the fellowship here in integrative medicine. But even perhaps more importantly, he's the founder of the integrative medicine programs. So over to you, Andy.
Thanks, Lee. I have the pleasure of introducing Dr. Lauren Hablitz. She's a circadian biologist and principal investigator of the Hablitz Lab and an assistant professor in the Department of Neurology, Center for Translational Neuromedicine at the University of Rochester Medical Center in Rochester, New York. She earned her BS in biology from the Pennsylvania State University in 2010. And in 2015, Dr. Hablitz earned a PhD in cell, molecular, and developmental biology from the University of Alabama, Birmingham, from the lab of Dr. Karen Gamble, investigating conserved molecular mechanisms of non-photic entrainment. After completing a postdoctoral research position at Oregon Health and Science University with Dr. Charles Allen, understanding the role of astrocytes in modulating cannabinoid signaling in the suprachiasmatic nucleus, Dr. Hablitz joined the Center for Translational Neuromedicine at the University of Rochester in 2017. Together with Dr. Micah Nedergaard, Dr. Hablitz defined the endogenous circadian rhythm of the glymphatic system and discovered a new rhythm in CSF distribution between the brain and the body. In 2020, she earned the Society for Research on Biological Rhythms 2020 Merit Award. She's a 2021 recipient of the Sleep Research Society's Outstanding Early Investigator Award for Circadian Control of Brain Glymphatic and Lymphatic Fluid Flow, which was published in the journal Nature. So thanks so much for joining us today. It's wonderful to have you.
There we go. Hi, everybody. Thank you for the kind introduction and for the welcome to be here today. As they so eloquently described, I am a circadian biologist, so I ask a lot of questions about timing and when we do things. But today I'm going to switch that a little bit and talk about sleep and why we sleep and how our lab views sleep. And so what you'll actually see on this screen here is a picture of a mouse brain where we just took the skin off the top of the skull and put dye into the cerebral spinal fluid, and you can see it goes really nicely up the middle cerebral artery. And this is an anesthetized mouse that is fully alive under a microscope. And so what our lab has really been interested in is how does this system that enables cerebral spinal fluid into the brain work? In what conditions is it impaired, and how might we fix that? And so normally I start with a big question, and that's why do we sleep, which I think fits great with this symposium talk right now. Sleep is evolutionarily conserved. It's flies, it's mice, it's my creatures on the bed, and it's literally everyone that's in this audience. But as far as like the sleep biology side of things, the first speaker did a wonderful job of saying reasons why we might sleep. What my lab and our research group focuses on is we may sleep to clean the brain. Being awake is extremely energetically costly. We have to interact with the world that we live in, and our neurons get tired too, and they start producing waste just like our muscles, but if we overexercise them. And so we need a way to get that out of the tissue. And so, working with Dr. Nedergaard, there's this hypothesis of the glymphatic system. So in the periphery, if you do exercise a lot and your muscles hurt a lot, the lymphatic system takes over and pulls the lactic acid from the tissue. But the brain doesn't have the lymphatic system. Instead, what it has is a thing here on this screen. You'll see blood vessels that are red to blue. That's arteries to veins. That's how blood travels. But what you should also notice is that in the brain, there's actually spaces around the blood vessel, the perivascular space, where fluid can actually fit down. And so these are actually kind of homologs to how the lymphatic system works, because once the blood starts pumping, cerebral spinal fluid can get pushed into the tissue to help clear all the waste from the neurons and ultimately pull it down the perivenous space to the periphery. And so I'll say the perivascular space has been known since the 1800s. Actually, the hypothesis that this worked well in sleep has been suggested since like 1890s. The biggest problem is it's really hard to see these spaces, right? Because you're looking at clear cerebral spinal fluid going into an organ that's surrounded by the skull into really small spaces. And so, the topic of perivascular spaces came up in the 1890s. It came up in the 1930s. It came up in the 1950s, the 1980s. And every time people are like, we don't know what to do with this yet because we don't know how to see it and how to look at it. Until Dr. Nettergaard's beautiful paper. And so the seminal study that they did was they trained mice to sit underneath a microscope. We use 2-photon microscopy. We can get pretty deep in the tissue, which, considering we're trying to image in a brain surrounded by a skull, this is really impressive. The two people, Hung-Yi and Lulu, that did this experiment actually trained these animals to sleep or to wake underneath the microscope naturally. They put dye into the cisterna magna, which is this little purple needle on the screen here, putting it into the cerebrospinal fluid to actually be able to travel through the brain. And this is the images they got, right? You'll see blue is actually blood vessels. You'll see two conditions here. You'll see wake and sleep. You'll see a green tracer and a red tracer when I start the video. And what I hope you can appreciate is in the red video, a lot more red tracer gets into the image screen and starts lighting up those blood vessels than during the awake state. And so this was really the first evidence where someone could look in the brain in real time and say, wow, cerebrospinal fluid and dye in cerebrospinal fluid can get really well into the brain when we sleep. They followed it up with another study that Dr. Jane presented really nicely, where they could show that radio-labeled tracer got out of the brain when we slept. So not only is it fluid going in, but it's also fluid coming out of the brain that increases while we sleep. Everyone might be thinking, okay, I mean, maybe you don't think about sleep deprivation, but this is about sleep. And so we want to know, if we lose sleep, does the system get impaired? But the other question is, does this happen in humans? And so there was this great study by a group of Norwegian MRI biologists. And what you'll see here is 3 different sections of the brain after gadolinium infusion of tracer after a patient was awake or allowed to sleep for 24 hours. And so here is the patient able to sleep. Where it's red, there's dye in the brain. Where it's blue, there's less dye in the brain, or radio-labeled tracer, sorry. What I hope you can appreciate is there's a lot more red in the sleep-deprived brain of the patients that had to stay awake for 24 hours. And so this study was really important because it was the first time, first of all, we showed that if you lose sleep, the system gets impaired. But second, we can see this active in real time in a patient population. Dr. Ida and Ringstadt's group has continued this work. It's really fun watching them go. A lot of their stuff has to do with, okay, how does this change in different disease models? They're really interested in normal pressure hydrocephalus and stuff like that. But where are we taking it? Why am I telling you about this story today besides sleep deprivation? One thing I want to introduce is the idea that sleep is important, not just to clean the brain, but also to help keep our brains and ourselves healthy. And one great example of that is the relationship between sleep and pain. And so what I hope you can see here is that this research group kept animals awake for different periods of time. And after they kept them awake for 6, 9, and 12 hours, they started doing testing on pain sensitivity. And so here you'll see hot play withdraw. If you keep a mouse awake and put it on a heat pad, it'll put its paw on the pad and it'll go, oh crap, it's hot, and pull it back. And you'll notice that in the red group, the one that had been awake for 9 hours, they pull their paw back faster because they notice it's hot faster. So this is increased thermal sensitivity to pain. They also did a von Frey test. And so this is if they take a mouse paw and they start poking it, how fast does the mouse realize it's being poked? And what you'll see is the red line is above that white line again. It means that it really takes less for these animals to realize they're being poked and realize that they're in pain compared to a group that's been able to sleep just fine. And then the final thing they did was inject capsaicin into the paws of these mice. So if you guys have ever cut jalapenos and you notice your hands start burning, that's because of capsaicin. It's a spice protein that ends up turning on pain receptors in your fingers. And so this happens in mice too. And what they notice is that the sleep deprivation group starts getting really, really irritated more than the group that slept. And so now we know the brain is cleaned during sleep. We know that if you lose sleep, you're not able to clean the brain. And we know that if you start messing with sleep, you start changing how you see pain. And so how do we put all of these things together? What is the connection between pain, sleep, and glymphatic function, right? We started testing this, and the first thing we just asked was, what happens if we do a chronic injury model in a mouse. How do they respond to pain and what happens to the glymphatic system, right? Overall, here's how we think it works. We would cause tissue injury and inflammation, which would keep the animal awake, which would impair glymphatic clearance. That's all pretty straightforward. But then the brain has to start responding to the fact that the waste isn't getting cleared from the tissue. Like your brain is dirty, and so what ends up happening, you start getting an immune response, your astrocytes start reacting with gliosis, and your neurons start noticing that stuff is wrong, and so electrical signals start getting remodeled. And that's when you start getting chronic pain. Once, even after the tissue injury is healed, your body can remember it was in pain, and we know that pain can cause sleep disruption. But this is a circle. So how do we start testing it? Because, you know, it's great to have a model and we're pretty sure that this works correctly, but where do we start teasing this apart? Is glymphatic function reduced in a model of pain? And so this is what we did. We use a spared nerve injury model where we actually cut the tibial and peroneal nerve in the sciatic nerve. And so these animals start experiencing pain in their back legs because we ended up trimming the nerves. What you'll see here is that same von Frey test I showed you earlier. This is plotted a little differently, but you can see once we give the surgery, the animals don't need as big of a poke to pull their paw back. And this can actually last for over 3 weeks. And so what we did is at 3 weeks, at that long-term point where the body is starting to heal itself, but the animal is still showing a pain response, we wanted to ask, how does dye get into this glymphatic system better? And so we did that same experiment before where we put dye into the cisterna magna, the cerebrospinal fluid pool. We let the fluorescent tracer sit for 30 minutes. We took brain sections and we looked to see how much fluorescent tracer got in with these animals that had a model of pain. And so here's what we find. If you're ever wondering what a coronal section of a mouse brain looks like, here's what it is. I hope you appreciate that there's a lot of white in this brain and they look like really linear pieces of white. That's because that's actually the perivascular space getting lit up by cerebrospinal fluid. And here's what the SNI brain looks like. And so there's this thing that happens where after 3 weeks of getting this spared nerve injury model of pain, glymphatic influx is declined. And, you know, we have a low sample size here, but this was also replicated in one of Dr. Nedergaard's preliminary works when we started this project. And so cool, we know pain can impair glymphatics. And so we go back to this model and we start asking, what can we do to actually restore glymphatic function? Can we reduce neuroinflammation to restore glymphatic function? We went in a different direction from what labs normally go into. Dr. Nedergaard had a beautiful paper that showed that if you give acupuncture to a mouse in its leg at ST36, the animal actually has an acute adenosine response that causes a local change in immunoreactivity that ends up releasing pain or reducing pain. And so our question was, if we start doing the same acupuncture technique locally in the same section, do these animals still have a change in glymphatic function over time, or can acupuncture decrease enough of that immune response to start recovering glymphatic function? And so we did the same test where animals had the SNI surgery. What I hope you notice this time is we don't do the experiment until 36 days after the surgery. So by now the animal actually is healing from this. Instead of the 21 days I showed you earlier, we looked at how well dye got into the brain. And this is what we found, which is a little messy, but that's okay. That's science. That's how we deal with it. The first thing you notice is that something happens at 36 days where we have increased variability in glymphatic function, but it's not as decreased as it was at 21. So there may be a time frame of when the brain is actually decreased and how it deals with that. We're looking into it. But the other thing I really want you to appreciate is that especially in the SNI group, we get an effective acupuncture and it recovers glymphatic function. And so now the question became, is this real? Is this a stress response of the animal? What is acupuncture actually doing here to the glymphatic system? And so now we're going to switch gears a little, right? We looked at, okay, pain can impair glymphatics. Acupuncture may be able to recover it. But now we just need to know if this works in healthy animals, or is this a combination of like stress and sleep deprivation and glymphatic dysfunction and pain that's causing a weird response when we give acupuncture on top of that in animals. So can this work in healthy animals to promote good glymphatic flow? And so we did 2 different things in this experiment. You'll notice we gave acupuncture for 2 weeks again, but these are healthy animals, so we say day 1 to 14. We used that same point in the leg that we were using before, and we also used a control one in the foot. So this way we know if it's specific to the one that actually feeds the lymphatic system that's near the part in the sciatic nerve that would get damaged, or if this is just us sticking a mouse in the foot and it getting mad at us. We did that same experiment where we put dye into the cerebrospinal fluid and let it go into the brain, and we found absolutely nothing, which is actually pretty cool because if you go to talk to acupuncture researchers, acupuncture doesn't always work in healthy individuals. It actually needs to have a it needs to have an inflammatory response to actually be able to work. And so our data actually kind of proved this in that in a healthy animal, acupuncture doesn't change how the brain functions. You know, it may have an effect locally, that's what Dr. Nettergaard shows, but the combination between the periphery interacting with the brain is disconnected if the animal is healthy. Okay. But I am a circadian biologist. This acupuncture was a fun, different way to think about how the periphery could interact with the brain. And so I wanted to get back to timing, right? We talk about timing of sleep, but we also have timing of a bunch of other things too, like eating. And the circadian field especially is really interested in how does giving food at the right time of day change how the brain works, and how does food change how disease works. And so we just asked a really simple question of if we give food when the animal should be awake compared to if we give food when the animal should be asleep, how does that actually change how the brain works, right? Does it change glymphatic function? And so what you'll see here is we gave 8 days of restricted feeding. And then we did that same glymphatic influx that we saw before. And here's what it looks like. So here is the control animals where we had normal amount of glymphatic influx. Time restricted feeding actually increases how well food gets into the brain. So when we give food when it should be there, dye gets in better. And that could be because if you think of a sine wave across the day, sleep would beef it up on one side and food may tamp it down on the other. So the amplitude of that is bigger. And so when we start looking about how well glymphatics works, if we give food at the right time of day, we end up getting more glymphatic influx. What's even more interesting is we started to ask if we screw up rhythms, do we end up screwing up the glymphatic system? And right now, it doesn't necessarily look like it. We gave food when the animal should be asleep. This is normally not when they eat. For mice, if you give them food when they should be asleep, they binge eat for like 2 or 3 hours and actually deprive themselves of sleep, and then they crash. And their whole activity cycle delays like 2 hours. But we don't see that reflected in the glymphatic influx. If anything, we're starting to trend toward an increase like we do with the restricted food during the active phase. And so one of the main goals of my lab going forward has really been thinking about how do we reconcile disrupting rhythms hasn't changed influx with some of our genetic models where if we do it genetically, if we break time in a cell, we start getting glymphatics unchecked. And so now we're trying to tease apart what is this sleep deprivation and sleep need part of how the brain clears waste versus what is the timing need of how the brain clears waste. Okay, tangent, it's fine. And so what have I told you guys about this far? This went a little faster than I thought it would. I am terribly sorry. But basically, we know tissue and injury and inflammation causes acute pain. We know that ends up depriving the animal of sleep, which can impair glymphatic clearance. And we know that ultimately, if you impair glymphatic clearance, you end up triggering a neuroimmune response that increases cytokine productivity, changes excitation of the neurons, and ends up with maladaptive circuit remodeling that can influence chronic pain phenotypes. And we know chronic pain can impair sleep, especially if you look at epidemiological data. Most pain peaks in the early evening, so patients have a big problem getting to sleep, and then patients with back pain end up waking up really early because they're in pain. And so what did I tell you today in terms of data? The glymphatic system cleans the brain during sleep. Dr. Nedicard beautifully illustrated that dye can go into the brain and radiolabeled tracers can come out of the brain. Paraida's group showed that if you sleep deprive a human, you can actually have retention of gadolinium dye in the brain. If you look at the work by Laura Lewis and colleagues, they're showing really nicely that cerebrospinal fluid kinetics across the brain actually change a lot during sleep. And it might be how the blood flow is interacting with the brain, which is another connection between the periphery and how the brain handles stuff. If you give sleep disruption, you decrease glymphatics and increase pain. And similarly, if you cause a chronic model of pain in mice, we start to see glymphatic function fail. And then finally, we may be able to have a peripheral reduction in inflammation help clear what happens in the brain and increase glymphatic influx. But this is only in animals that have had S&I. We don't see this in healthy animals. And then finally, you know, sleep and circadian rhythms end up interacting. And if you help circadian rhythms, maybe by giving the animal food, you can also increase glymphatic function. Probably because if you restore the rhythm correctly, the animal is also sleeping the way it should. And so what do I want you guys to really take home from this whole little spiel that's a little bit different from what we've seen so far? One thing that's really interesting about the glymphatic system is pretty much any disease model you look at has an impairment of glymphatic function. And so we have a couple listed here. Things like aging and things like inflammation have been pretty well shown to decrease glymphatic function. These other ones are kind of question marks. We know respiration can change how well cerebrospinal fluid gets out of the skull, but we don't really know how that affects the brain tissue. We know that patients with depression may have impaired glymphatic function, but we don't really know why. And there's a huge push to understand how the cardiovascular system in the periphery interacts with how the glymphatic system works in the brain, because a big part of the glymphatic system is hijacking the blood vessels to push in cerebrospinal fluid into the brain. One thing my lab has really been doing is looking to see how changes in glymphatic function after stroke may be regulated by cytokines. And this inflammation may change how we see pain, which is a whole different thing, which can impact sleep quality. And so the take-home message of today's talk is really, we need good sleep to keep our glymphatic system in order, but also if our glymphatic system starts failing, that may drive how we sleep. And I know I ended a little bit early. I'm terribly sorry, but I want to say thank you, especially to the people that helped with the S&I and acupuncture work. Those are highlighted in orange, and my collaborator, Dr. Neddergaard. I wanted to thank my funding sources, and most of all, thank you for your attention.
Thank you very much. It was an excellent talk. And just wanted to ask a quick follow-up question, since we do have a little bit of time. One of the trends I think we've been talking about during this is that we have sleep deprivation being bad. When I first heard about the glymphatic system, that was kind of like an aha moment for me. Do you think that maybe this is driving a lot of those relationships, and maybe give us some thoughts as to how we can remedy that?
Yeah. So I think Paraida's group showed beautifully that if you sleep deprive a human, the glymphatic system tanks. And there's been a lot of literature that shows if the brain starts getting healthy, the glymphatic system tanks. My question recently has been, is there a continuum to glymphatic function? Because this is how the brain cleans itself. And you can imagine if this fluid plumbing system is going too fast, it's going to start pulling the tissue with it. And so one of the things, in terms of sleep deprivation, yes, sleep deprivation is going to impair our system. We may be able to fix blood flow to fix that. We may be able to, you know, manipulate some like peripheral stress responses to fix that. But I think the field has a lot to do in terms of when do we start seeing glymphatic dysfunction, especially in terms of chronic sleep disruption. And is it always a downhill, or is it like a peak that troughs? I hope that was an answer.
Yeah, absolutely. And I think this will probably be relatively new to many of our listeners, unless they listen to the podcast episode that we did with Dr. Jane. I could give them a bit of a sneak peek, just because one of the things we talked about in the podcast is that a lot of people just, and even in my family, I've heard this statement, I'll sleep when I'm dead. And I think we're realizing that that's not at all how this works. Like, you need to sleep so you can live and enjoy the life that you have. And hopefully this will help people prioritize sleep.
Yeah, definitely. And I'll say, the UK has actually started, or not the UK, Europe, some places in Europe have actually started putting protections in from shift work, and actually have been giving hazard pay for shift work, because of the idea that we kind of need sleep to survive.
Yes, it's clearly quite important. All right. Well, thank you so much.
Thank you.